Good morning and welcome everybody. Here's the Professor Pallavi Gule from Kekewag Education Society. Today we will go for the new topic that is about the seed dormancy. Content of the topics are introduction about the dormancy, classification of the seed dormancy, mechanism of dormancy, factors causing dormancy, methods to overcome the dormancy and biological importance of seed dormancy. This is the typical structure of the seed: cotyledon, epicotyl, radicle, testa, endosperm, and eleutheron layer. Now, the introductory part about the dormancy. During the developmental cycle of the plant, at some phase, certain structures like buds, tubers, seeds go through a period of temporary suspension of growth activity. Such state is called as a dormancy. In plant physiology, dormancy is the period of arrested plant growth. It is a survival strategy exhibited by the many plants under unfavorable conditions. To overcome unfavorable condition, lower plants produces endospores, zygospores and oxospores, etc. Now, what do you mean by seed dormancy? Seed dormancy is defined as a state in which seeds are unable to germinate even under favorable environmental conditions normally provided for germination. Now we will go for the classification of dormancy. Seed dormancy is generally classified into two types: primary dormancy and secondary dormancy. Now, what do you mean by primary dormancy? Primary dormancy is due to the internal causes even if all external environmental conditions are duly present or favorable for the seed to germinate. Secondary dormancy is when dormancy caused due to the external factors like absence of sunlight, CO2, or very high or very low temperature conditions. Classification of seed dormancy: exogenous dormancy and endogenous dormancy. Exogenous dormancy is physical dormancy, mechanical dormancy, or due to the chemical dormancy. Endogenous dormancy is due to the physiological dormancy, morphological dormancy, or the combined dormancy. Now, physical dormancy. Dormancy that is caused by an impermeable seed coat. It is the result of impermeable layer that develops during the maturation and drying of seed or fruit. It prevents the seed from taking up water and gases. As a result, seed is prevented from germinating until dormancy is broken. Generally, physical dormancy is the result of one or more palisade layers in the fruit or seed coat. Mechanical dormancy occurs when the seed coat or the other covering are too hard to allow the embryo to expand during germination. Chemical dormancy is due to the presence of inhibitors within the seed coat itself and which causes the seed dormancy. For example, cottonseed pericarp contains abscisic acid, coriander seed pericarp contains coumarin, or the paddy hull contains abscisic acid. Physiological dormancy. Physiological dormancy prevents the embryo growth and seed germination until the chemical changes occur. Photodormancy. Light sensitivity affects the germination of some seeds. These photoblastic seeds need a period of darkness or light to germinate. Thermodormancy is a seed sensitivity to heat or cold. Some seeds germinate only at a high temperature. Morphological dormancy. In morphological dormancy, the embryo is underdeveloped and undifferentiated. Combined dormancy is seed have both morphological and physiological dormancy. Mechanism of dormancy: hormonal control theory, temperature and gaseous exchange restriction theory, and metabolic control theory. Now, what do you mean by hormonal control theory? Hormonal control theory is dormancy causing hormones are ABA and ethylene. Dormancy causing hormones and dormancy provoking hormones GA and cytokinin ratio determines the fate of dormancy. Chilling treatment light exposure increases gibberellin level in seed and causes germination. Temperature and gaseous exchange theory is restricted oxygen uptake due to the hard seed coat, and high temperature induces dormancy in seed. Oxygen deficiency leads to inhibition of the cytochromin oxide activity, which leads to dormancy. Metabolic control theory. Dormancy in seed is controlled by metabolic energy produced in seed. During dormancy, respiratory system blocked, ATP synthesis is blocked. Increase in AMP and DAP leads to the increase in activity of the glycolide pathway. Production of the growth inhibitor at a lower concentration of oxygen. Factors causing dormancy of seeds, that is hard seed coat dormancy due to temperature, immature embryo, light sensitive seed dormancy due to growth inhibitor chemicals and excessive salt. Dormancy due to hard seed coat. Testa of some seeds are hard and multilayered. Hard seed coat prevents germination due to the following reasons. Hard seed coat prevents the entry of water into the seed. Hard seed coat obstructs gaseous exchange, especially of oxygen. Oxygen is necessary for respiration. Hard seed coat causes a mechanical restriction, thus radicle does not come out. Dormancy due to temperature. Some seeds require chilling treatment for germination. For example, apple, walnut, and peanuts. Such seeds need chilling temperature, that is from 1 to 5 degrees Celsius for few weeks. Immature embryo. Embryo is not matured at the time of harvesting. For maturity of such embryo, resting period is required. Light sensitive seeds. There are two types of seeds for light sensitivity. Positive photoblastic seeds. Seeds of some species like lettuce remain dormant in the dark. They germinate when they are exposed to light. Negative photoblastic seeds of some species like Nemophilia phylla and silence remain dormant when exposed to light. They germinate only in the dark. Dormancy due to the growth inhibitor chemicals. There are several naturally occurring compounds which inhibit the germination of seeds like coumarin, organic acids, aldehydes, alkaloids, and the fluorides. Example, tomato fruit pulp, caffeic acid, and ferulic acid, which induces the dormancy in tomato. Methods to overcome seed dormancy. Mechanical scarification, impaction, cold stratification, removal of inhibitors, treatment with growth hormones, specific light requirement and pressure. Scarification. Any treatment that is physical or the chemical that weakens the seed coat. Rubbing or puncturing. By rubbing them against a rough surface or puncturing the seed coat with pointed needle. Chemical treatment includes treating the seed with a strong acid and organic solvents such as sulfuric acid. Impaction. It is the process of shaking a seeds to clear blockage of testa pores. Stratification. Treatment of the seed. Treatment of seed at a low temperature under moist condition. During this anatomical and biochemical changes that take place in the seed. Growth promoting hormones increases and growth retarding hormone decreases. Removal of inhibitors. Growth inhibiting chemicals removed by repeated washing with water. Treatment with growth hormones. Gibberellins substitute the cold treatment requirement in many seeds. Cytokinin promotes germination of many seeds. Pressure. Dormancy can be broken by exposing the seeds to high atmospheric pressure. Specific light requirement. Dormancy of photoblastic seeds can be overcome by light exposure. Red light promotes germination in lettuce seeds. Biological importance of seed dormancy. Dormancy allows the seed to remain in suspended animation without any harm during drought, cool, or high summer temperature. The dormant seed can remain alive in the soil for several years. It helps the seed to get dispersed over a long distance through unfavorable environment or inhospitable area. Dormancy induced by the inhibitors present in the seed coat is highly useful to desert plants. The seeds germinate only after a good rainfall which dissolves away the inhibitors. It follows storage of seeds for a later use by man. This was all about the seed dormancy and methods to overcome the dormancy. Thank you.